I guess we can get started. So today we are going to talk about environmental sampling. So we have already do the sampling yesterday, so I guess we have some experience. So first, I would like to talk about the importance of environmental sampling. And then I will go through some of the process during the sampling. And the first is about planning. And then we are going to collect and transport our sample. By having a sample, we cannot analyze the sample directly. So we need to process the sample. And after the processing, we need to do the sample analysis. And finally, we are going to do data analysis and data processing. So I'm going to talk about them one by one. But before we get started, I would like to talk about some of the backgrounds. And so for environmental engineering, we are interested about the pollutants in the environment. And the pollutants mainly are distributed in 4 phases in the environment. It can be in the water, in the soil, and in the air. And also the pollutants can exist in biota. We can say in the biota, we have the animal and we have the plants. So basically we have these 2 components in the biota. And for the pollutants in the animals or in the biota, we usually care about ecological risk. And I will be talking about this in the organic chemistry unit. So basically we are interested about how to sample the pollutants from this medium. And this is the problem of from sample to data. So if we want to get the data from a sample, we need mainly do these 6 steps. And the first one is planning, which is about what we are going to do and how we can do that. The next one is about sample collection. And the next one is about sample transportation and preservation. Then we have the sample processing, because for most of the samples, we cannot analyze it directly. We need to do some process. And then we can send the sample to the instrument and do the analysis. And finally, we get the data. We may not directly use the data. We may check or process the data before we get the conclusion. And then, so in this class, I'm going to talk about them one by one, but some of them will be focused and some of them will be less focused, and we will be talking about them in the future lectures. And so for this one, do you guys have any questions? No? Okay. So first is about planning. So before we start our project, we need to have a good plan. And although it is a good plan, it says very easy, but actually it is not quite easy. So we need to mainly consider 4 components when we are doing the planning. The first one is about experimental design. Like we need to think, what data will we collect, and we use any pre-existing data or do we need to do any site selection? And in terms of the lab that we do yesterday, we can see we are going to collect the soil data, especially like the soil humidity, I think. And then we are going to measure the organic pollutant concentration in the soil and the microbial in the soil. So these are the data that we are going to collect. So will we use any pre-existing data? I would say yes for our lab, because when we get the organic pollutants data or microbial data, we are using our data to compare with the soil data from other sites. So we are going to use other pre-existing data. And for the site selection, actually, when we are designing this lab protocol, we selected the site because we want to make sure we have the soil that has relatively large amount of microbial that we are interested in. So I think when we are doing the sampling, we take the soils from the bioretention cells and something around that. And the next one is about documentation. Like we need to have the log sheets, field notebooks, and lab notebooks that they have. We also need to do a change of custodial, but for us, we do not do that. But for Maria, she will be managing all of these stuffs and to make sure we can get everything that we want. Another one is standards opposition procedure or SOP. So basically our lab protocol is the SOP, and I will talk about this in a little bit more details in the next slide. And the next one is about methods. When we are going to measure the concentration or do some other measurement, like the method is important. And usually we have three options. The first option is to use EPA method. So basically EPA means Environmental Protection Agency. So this is the regulatory government agency of United States. So EPA methods usually represents like the governmental files or the regulatory methods. And we may also use the literature methods, like the method from literature. And here we may need to make the choice. Which one should I use? So about these two methods, do you guys think which one do you guys prefer? Like if you are doing the selection, which one do you prefer? Do you guys have any ideas? Yes. Yes. And any more like the literature? You?
I guess it depends. We'll have to see if the EPA procedure fits what we would like to research on, or would still adhere to what we want to preserve, or the procedure is also existing in EPA. Sometimes it's not.
Yes, I would also say it also depends, but for me, I would say EPA methods is preferred because it is an EPA method, which means it is well documented, and many other people they use the same methods, so it is supposed to be work. On the other hand, the governmental file is scientifically defensible, that is correct, but it is also legally defensible. So if I use this method to do the measurement, but some other people, they are not agree with my results, they may have a lawsuit on me. Then I say I use the EPA method, and then it is more legally defensible. But for some of the situations, because if we are doing the research on new stuffs, then we may not have EPA methods. At that time, we may choose from the literature because the literature usually have the novel method for new stuffs. And the last one is about supplies, which is about sampling suppliers and containers and the transportations. And in lab 4, again, I think Maria was thinking about all of this and she was in charge of this. But on this side, we know that based on our daily life experience, we know that plants can't keep up with changes, which is very sad. So here I want to say that even though this will happen, but we will still have plans. And when we have some problems, then we can just go back and to revise our plan. And during this back and forth, actually the experience of us will increase. So we will be more experienced about making a plan. So as an experienced planner, we can have the plan that cover most of the unexpected situation. So I would say experience is very important for engineers. So more about documents. On the left side, I just find some lab notebooks from the Nobel Prize laureate. And we can see the note is very clean and I like this style. But for my notes, it was that's like this. Okay, anyway, so we are going to do the lab notes, field notes, and chains of Clostridium and SOP with version control. And I would say version control is very important because as we are doing the experiments, we may find some methods may not proper and we may make some adjustments. So we may have different versions of SOP. And you guys can see for the lab protocol as the last page of the lab protocol, we have the version control note. And another one is we need to avoid we need to have unambiguous sample ID. We just make sure the sample ID is recognizable. and it is kind of unique. So all of this process is to make sure, is to keep notes, but is to record what we have done. But actually, when I was thinking about this, I'm not quite satisfied that we know that for now, when we are doing a documentation, especially for experimental process, we write them down in a file and we write down everything. But I think nowadays we have some other technologies, like we can record the experiment process, or actually we can use like the histogram to record the process. Because like based on my experience and experience of my friends, when we are trying to follow a lab protocol, the description to do the same experiment, many times we will fail. And there will be some other unknown factors influence our experiments. So if we can record the whole process by the video or the histogram or some other VR stuffs, then sometimes we may find some other factors that is not written down in a file, but it exists in the environment. But I do not know why people does not do this. Maybe it takes a lot of time, but I hope in future we can have more advanced methods for the documentation. Okay, anyway, so more about SOP. SOP refers to the standard operating procedure, and this is a set of written instructions that document a routine or repetitive activities followed by an organization. And it is important because it can provide clear information and instructions, and it is consistent so that we can make sure different people at different places, they are doing the same experiments, they can have the same process. And it is also related to quality assurance and quality control, which I will be talking more about in later slide. And here shows the common structures or the contents of the SOP. Like usually has a title page, a table of contents, procedure, which is the most important one, and quality control and quality assurance part, and some reference. So I think for assignment 5 or assignment 4, you guys will be working in a group to develop a SOP. And on the right side is a sample SOP, and you guys can find more samples on the Canvas page. So this is the SOP of my project, which is the remediation of DDT, which is an organic pesticide. And so we can see this is a title page, and you guys can just take a look at this one from Canvas and see what it looks like. And so for SOP, do you guys have any questions? No? Now we have a plan, and then we bring all the stuffs and go to the field. The first thing we are going to do is sampling. And in terms of sampling, the most important thing is to find the representative samples. In terms of representative samples, for the object, it usually have some properties like heterogeneity, like if the object is heterogeneous or homogeneous. We know that for homogeneous sample, it is relatively easy. We can just take any parts of the sample and the concentration or some other properties are expected to be the same. But for the heterogeneous samples, it is relatively hard to find a representative sample. For example, this shows the pH value of a site, and here is a scale. We can see that if we want to find some representative samples on this site, then we need to find a good sampling plan, a plan with a limited number of sampling sites. We want to make sure we sample the site with larger concentration and the site that has the lower concentration, lower pH. Another one to make sure we have representative sample is to do replicates. So again, for this site, if we only do one sampling, we can have one pH. But if we do some other replicates, we can find the pH will be different. Then we can have some general ideas about the variation or the variability of the pHs in this place. So for this one, do you guys have any questions? No. And now let's look at a example. So we have many candies. So I have a question. Do you guys think if we consider these candies as some sample and we are interested in the color of these candies? I know the flavor of this should be the same like the chocolate, but the color are different. So are these samples heterogeneous or homogeneous? So how many of you guys think that is heterogeneous? All of you? And how about you guys? If you guys think it is heterogeneous, you can type 1 in the comments. Or if it is homogeneous, you guys can type 2 in the comments. Okay. So I think I asked this question because the answer will not be that simple. That's why I asked this question. So let's suppose, okay, no one reply. Okay, that's fine. Okay, let's suppose we can pick up the candy, one candy at one time. So maybe first time I get this one. Yeah, heterogeneous. Yeah, thanks for your reply. Then we may get this one, and then we may get this one, and this one, or this one. So we can see if I can pick up the candy, one candy at one time, the color is expected to be different. And then we can say this is heterogeneous. But suppose I have a large spoon and my sampler, only available sampler is a spoon, and it can only take like a chunk of candies, like here. So this is first sample. This is second chunk of sample, and we may have the third chunk of sample. And we can imagine, because the candy is supposed to be well mixed, and in this case, for every spoon of the candies, we would expect the average color will be similar or the same. Does that make sense to you guys? So in this case, the sample is homogeneous. So in this case, I want to show the point that if we are interested in if we are going to identify if the sample is homogeneous or heterogeneous, it depends on the method that we are taking the sample. So if we go back to this site, we can see this is 2 kilometers. And based on our daily experience, we know that for most of the sample, that will be just 1 meter or around 1 meter. So this is heterogeneous for sure. But let's just say if this scale is micrometer. So this is a pH distribution of a like soil in very good resolution, and the scale is only like about the micrometer. And when we know that when we are taking soil samples, the size of the soil sample may just about several centimeters or 1 meter. And in that case, even though the pH changes inside this very small chunk, but for the large soil, the average pH may be the same. So this may also be homogeneous. So, so the point is that it depends on the sampler. Okay. So do you guys have more questions about that? No? Okay. I didn't find my water, but that's fine. Okay, let's talk about the exact sampling or samplers. So first, we are going to sample water. And for my research, I didn't sample any water samples. So I guess maybe some of the graduates here, you are more experienced about the water sampling. So for the liquid samples, we have like different types of instruments. And actually for some of these samplers, I have never seen those before based on my career. So I just searched for some of these samples. And from the right side, we can see this is the bacon bump sampler, which is like this. So I guess it can absorb the water samples into this. But I think the main focus is that this sampler is very expensive. So I was like this when I find this sampler. And we also have like the drum sieve sampler, which is like this. So we can get samples from the drum. And we also have like the deeper sampler or some Wildo acrobatic sampler. Have you used that before? I think Amir is working with the wastewater treatment before.
So wastewater is actually fine. I think this might be for lakes where you want to measure different depth of the lake.
Oh,
I think for wastewater is pretty homogeneous most of the time.
I see. Oh, because they are
Well
also
mixed.
mixing.
There's a mixer and
Yeah, that makes sense. And usually the water must be representative of the body of the water from which the sample are collected. So basically they should be representative. And usually we will fill the container to within 2 centimeters of the cap and close the container. And I put a star here because I think the general idea is that when we are collecting water samples, we want to make sure the volume of air is minimized. So we want to avoid we have the air in the sampler, because if we have the air in the sampler, then when we have some chemicals in the water, they will be escaped from the water solution and to the air phase. And in that case, the composition of the water sample will change, which is bad. So we want to minimize the volume of the air phase. Also, we have some parameters like the pH or the temperature of the water. They should be measured at the site of collection. And again, because the constitute or the position of the water sample may change, so the pH may also change. And again, and also the temperature. And the depth of the sampling and the appearance of the water sample should be noted. So like some on-site characteristics of the sample should also be noted, because when we get them to the lab, that may change. So any more questions about water sampler? No. And then the next is about soil sampling. So we have already done that like yesterday. So I think we have more. So for the tools, I think yesterday we used the shovels because we take relatively large chunk of the soil, but sometimes we may use the small shovel and the tiny shovel and the ice cream shovel. I just call them like this. And we have some other instrument. And here is the soil core sampler, I think, which is also frequently used when we are interested about the depth of the soil at one location. And so for the soil sampler, for your soil sampling, we need minimal field preparation and not preserved. So sometimes the soil sample will not be preserved. We may only preserve a small amount of sample. And again, the soil may have some, because the soil may be heterogeneous. So before we collecting the soil sample, we may try to homogeneize the soil sample. For example, we have a site and then we sample all of the soil from this place. And we know that even though at this small place, the concentration of the pollutant or the pH may be different at different locations within this small region. So in this case, we collect the sample and then we may place it into the bucket. And then we mix it first. After mixing it, we put it into the jar. I'm not sure if you guys do this yesterday. No, just take the small sample into the jar. Yeah, okay. Yeah, but for some cases, we take a relatively large chunk of the soil, mix it, and then transfer a small amount into the jar. So the soil sample in the jar is guaranteed, like kind of guaranteed to be homogeneous. So sometimes, so we usually keep the soil in the dark with free access of air. And usually we analyze the soil sample within a limited amount of time, especially if we are interested in the microbial activities. And here it also says prior to the homogenization, we may remove some other stuff like the roots or leaves from the soil sample because we are only interested in the soil particles. So, and then we return the removed material to the sample location. So if we have extra soil samples, we just dump that into the original location. I mentioned this because for the sediment sample, this is different. So for the sediment sample, let's just go to this first. Do not discard any unused sample into the body of the water during the sampling event. And I will, so do you guys have any ideas why? For the soil sample, we can put it back to the original location. But for the sediment sample, we should not do that. Do you guys have any ideas about why they are different? So for the soil sample, let's just say this is the water phase. And this, so we have the sediment here. So for the sediments, they are like under the water. So there is usually no oxygen in the sediment. So in the sediment, we may have, let's just recall when we are talking about acidity, we may have the sulfur or some menthol, like sulfur salts. So like this M refers to the menthol, and we know that is this -2, and this is 0. And they are happy in the sediments because there is no oxygen to oxidize them. So when we put this sediment in the surface water, because we are grabbing it, and in the surface water, we have oxygen. So this sulfur, when they meet oxygen, oxygen can oxidize them into sulfuric acid. And in the same time, so basically the H plus will increase. So basically we will add acidity to the water body. On the other hand, we also have this menthol, and these menthols are usually like the heavy menthols, like PD or manganese or some other heavy menthols. And these heavy menthols are very soluble with sulfuric acid. So when we put them back, because they have already exposed to oxygen, so these heavy metals will be dissolved in the water body, and so which is bad for the environment. So in this case, we should not return the sediment. And for the sediment, for the sampler, we have the grab sampler, I think something like this. And we also have the core sampler, and we also have the traces like this one. This is pretty large. And also it depends on the types of material encountered and decontamination or receive the grab sample as required. So because if we have extra sediment sample, at first we need to decontaminate it before we return it to the environment. And so for this one, do you guys have any questions? And the next one is air sampler. So if we are interested in the chemicals in the air, we can use like the air sampler, like the gas collector vessels, and like the gas bubbler or bottle bag. So like this, I have used this before. So basically, before going to the lab, we are going to extract all of the airs from this bottle. So this bottle will be vacuumed, and then we just go to the place and open the regulator, and then it can gradually absorb air from the environment to the bottle. So again, and sometimes we will use the air filter, because if we are only interested in the gas phase, like, you know, in the air, we have the particulate matters and the virus or the bacteria, so we want to filter them out. And sample collection equipment must be assembled outside the contamination zone prior to sample collection. So again, we want to avoid contamination of our sampler. And record is usually the records are different, or I would say when we have some different characteristics or parameters for air sampling. So which is flux. So we know flux is a function of flow rate of the air gets into the sampler. And also it is related to the sampling period. And for my study, because I did some ammonia, but I was also looking at the particulate matters in the air. So I'm not sure if it is obvious, but we can see there are some smoke from this plant. And so they are the particulate matters. So to sample the particulate matter, we use some different samplers. So this is a sampler for particulate matters, or PM, on the side. And here is the structure of particulate matter sampler. So basically, we have some flow that's sucking air from here, and then we will have the air going to this path. And inside this structure, we have some small paths or some channels. In this case, we want to make sure the large particles, because of the inertial, they are big mass, they will be just thrown on the surface of this plate. But for the small particles, they have small inertial, so they can go through this path and then go to the filter. And here we are measuring PM10. So PM10 refers to the diameter of the particles, which is smaller than 10 micrometer. And I will talk more about this in the future unit. So in this case, then some small particles, they will be collected at the surface of this filter. And on this side, this is the appearance of a new filter. And after sampling, we can see we have some particles here. And because the experimental site is a cotton gin, like the cotton processing plant, so most of these particles are the cotton fibers. And we can see these are the cotton fibers, like very small cotton fibers collected. Yes.
So for the air, the sampler is on the ground, like it's close to the ground, but the pipes are still like blowing the air out like towards it. Like, if you go back one more slide.
Yes, ma'am.
Yeah, so the sample's on the ground, but that
Oh, yes.
it's still going up like into the air. That's like
Yeah, it may go up and it may go vertically. It depends on the temperature of the outflow.
okay.
But that's a good point. So in this case, we have the ground sampler, and we usually most interested about the ground concentration of the air pollutant because we are human, we at the ground. So, and usually this height is about 2 meters or 1.8 meter, which represents the height of an adult. But we can also say we have sampler towers here. So yeah, so on the back, we still have some other PM samplers. So, and then we sample the contaminate at different heights. So, yes.
Are there samplers in the actual not this picture, but other pictures? Yeah. Are there samplers in the plume where it comes out?
In the
They start.
here.
Yeah, are there samplers in the actual plume itself?
In here, like in the plume itself.
Oh no, it's my bad. Sorry, in the actual like structure.
In here.
Yeah.
Yes. Like this one. So we have some samplers because sometimes we are interested in the outflow concentration, so we can do the modeling at the source strength. So in this case, we have some other samplers here to collect all of the samples.
Okay.
Yes. Any more questions? No. And we may have some other samplers about the biota, and like we may use the swipe, and I'm not going to read them. And we also have the vegetation samples, like we can use some other different method. Okay, so you guys can read this after uploading this slides. When we get the samples, then we are going to think about sample transportation and preservation. And I just put the ice here because I think for most of the environmental samples, we just freeze it. Let's say for preservation techniques can retard the chemical and biological changes that inevitably continue after the sample is removed from the parent source. So we say there are some biological changes, but basically it is chemical changes. There are some methods of preservation, like retard the biological action and retard the hydrolysis of the chemical components and complexes, and reduce the velocity of the constituents, and reduce the absorption effects. So if we summarize them together, we can see that by lowering the temperature, all of this process will be slower. Like most of this process will be slower. So freeze the sample are usually the common methods to preserve the sample. But we can see that by using this method, something will change, like for the microbials, because for some of the microorganisms under the very low temperature, they will die. So we cannot use this method to preserve the microbial activity samples. And we also have like generally limited to pH control, chemical addition, refrigeration, and freezing. Okay, so for this one, do you guys have any questions? So here I want to show this paper, so this paper is from our group, and we can see the first author is Maria, and she was doing this, and also me. So we are doing this, and we can see this is an on-site evaluation of some pesticides in the soil. So that's why we developed this soil sampling protocol. And because we have done our own research in this paper, so we just modify our methods to what we have done on yesterday's lab. And if I'm interested, and this is the original method of our sampling, I guess this is a little bit too small. So you can take a look at this, and we just revise this original method to our lab. And the next one is about sample processing. So again, we cannot directly analyze our sample for most of the situations. So we need to process our sample first. Like we have some techniques like the drying, like to remove the water, or the filtration to remove some unwanted particles. And we can also do the extraction, which transfer the analyze into a different solvents or phases. For example, if we are interested in the pesticide concentration in the soil, and then we can do the extraction, we can extract all the pollutants from the soil into the organic solvents for further analysis. And I think we are going to do this in lab number 7 for the organic chemistry lab, and we are going to use the soil that we collected yesterday. For the water sample, we may also use the organic solvents to do the extraction if we're interested in the organic pollutants in the water sample. And I think in lab 7, we are going to use like this microwave extraction methods. Maria is still working on revising the methods, but most likely we are going to do this method. So basically we use the hexane to extract the organic pesticides in our soil sample. And finally, we can have the contaminants or the chemicals in the hexane solution. And after that, for most of the situation, we will concentrate our sample because the concentration of the pollutants in the environment is usually very low. So if we concentrate their concentration, they will be detectable. And after extraction, one can reduce the volume to concentrate the nanolytes. And for this one, you guys have any questions? No? Then after having the instruments, we are going to use the calibration curve to calculate the amount of chemicals or the concentrations that we are interested. And we have talked a little about calibration curve on statistical lectures. And here is the calibration curve. And in this case, the x-axis is the concentration and the y-axis is response or the signal. And again, they are interchangeable. But here we have a question. You measure a sample with a response of 6 blah, 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 which is this. And we have the similar question in the quiz. So we can see this response is larger than the range of the calibration curve. So how do you determine the concentration of your sample? So before answering this question, I want to talk about this slide first. So here I want to introduce two concepts. The first one is limits of detection, LOD. Another one is limits of quantification, LOQ. So let's look at the first one first. But before we're looking at the first one, let's just look at these three figures. So we know that when we are sending our sample to an instrument, the instrument will finally convert any information into the electronic signal or electricity signal, and it will be something like this. Like we have this like white noise from the environment, so it will be something like this. So if we have some sample like maybe at this time, and then because our sample can generate larger signal, so it will be something like this. And in this case, we can see that this is very different from the noise. So we know that we have some samples or have some chemical of interest in this place, but we can see this signal strength may be relatively small, like in this case. So it may be significantly different from other noise, and it may not be. But sometimes we will also have seen something like this. This is like lower than some other white noise. And in this case, we are sure that the signal is not significantly different from the noise. So in this case, we cannot determine if we have sample or not. But in this case, because this is much higher than the white noise, so we know that there must be some sample, must be some chemicals in our sample. And sometimes when we have this situation, so when we need to do some statistical analysis on this one, but this is not focus of this class. So basically, in this situation, we know that we have some sample, we have some chemical in our sample, but we cannot determine how much we have, because if we want to determine how much we have, we need to use the calibration curve. So for the limit of detection, this is the lowest amount of analytes in a sample which can be detected, but not necessarily quantified as an exact value. So we only know the signal is significantly different from 0. And we also have the limit of quantification. It means the lowest amount of analytes in a sample which can be quantitatively determined with suitable precision and accuracy. So in this case, we are going to use the calibration curve, because calibration curve establishes the relation between concentration and signal strength. But for the calibration curve, I would like to talk about this one first. So this one shows, so this amount almost we can consider it as a concentration. And this is a measure of intensity or the area or the peak or the signal strength. It can be anything that is from the instrument. So this picture shows the real relation between concentration and the signal. We can see that this is not a straight line. This is a curve. And from this plot, we can see that if we have very, very low amount of chemicals, the instrument is very insensitive to the change. And when the concentration is pretty large, the instrument is also very insensitive to the change. Only in something in the middle, and we can see it is approximately linear. So we usually construct the calibration curve in something like in this range. And in this range, we can consider it as linear relation. So suppose if we have the signal here or here, and we can see if we goes to here, the concentration will be like this. But if we extrapolate our calibration curve, because in this range, it is not linear anymore. So we may overestimate the concentration of our sample. So in other words, we do not know what is the concentration of sample when the signal is out of the range of our calibration curve. So in this case, we cannot determine the concentration. So let's go back to this question. You have a measure with a response of 6, blah, 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 and we can say it is here. If we extrapolate our calibration curve, then we may get a relatively large value. But we know that this curve may become like this, or it may be this, or it may be something in between. So we are not sure what will it be. So how can we deal with this problem? We generate two methods. The first method is to reconstruct another calibration curve that covers this range. So as long as it is still the linear relation, we can plug in the signal into the new calibration curve, and then we can get the new concentration. Another method is that because we know that it has a large signal, which means the concentration is supposed to be large, then we can dilute this sample. So we can maybe dilute into the half and then do the same test and say if it is in the range of our calibration curve. And in practice, the second method is preferred because we only have to dilute the sample and run the instrument. But for the first one, we know that when we are doing the calibration curve, it takes a lot of time. So most of people are not willing to do that. And so for this one, do you guys have any questions? No? Yes.
So for like if we were trying to look for like iron or like something specific, is there like set limits? Like you said that if we were trying to figure out like what that upper like extreme value was, that we could maybe take
This.
more data points.
You mean this one?
Yeah, but so I guess like think that for a given like for iron, say that it would always like start to level off, become nonlinear at the same time.
Yes,
Yeah.
like when it gets to a very high level, it will ultimately become nonlinear. Yes, and in practice, we know that for many of the cases, we do not know what's the concentration of our sample. So we do not know what the range should be for our calibration curve. And in practice, we may just read some literatures, or we may just try some tentative calibration curves and run some samples first and have some general ideas about what will be the range of our sample. And then we can come up with a final calibration curve. So any more questions? No. And the last one is data processing, and I think it has been covered in the previous lecture. And for the QA and QC, it will be discussed in the future lecture. But here I want to mention some general concepts. So basically, it will be discussed in details in lecture 13. Again, I'm not going to read this here, but basically, QA refers to quality assurance, and it is more about the process, like the process that we want to make sure our data is accurate and precise. And the quality control usually focus on the final products or the final results. So we are going to do some check about that. And here are some general concepts related to QA and QC. And we have like different parameters here. And again, I'm not going to read them in details. And you guys can read this and have some general ideas about this concept when I upload this slide. And we can see we have like replicates, blanks, spikes, and lots of them. So you guys can just read them and have some general concepts. So in lecture 13, I will show some examples for more details about Q and QC. And we can see for Q and QC, the main idea is to check the accuracy and precision of our methods. We want to make sure the accuracy and precision of our methods satisfy the requirements. So at that time, I will go back and discuss Q and QC in details. And that's the end of the class. But before the end, I have some final notes. So for next week, we are going to have Unit 4, Learn from Professionals, and that will be online only. So we are not going to class because the guests, they are only available online. And we do not have homework assignments for Unit 4, but we have quiz. And some students also mentioned that because for assignment 2, like it takes a lot of time. And one of the reasons is that we may have some repeated calculations for the error propagation. So here I want to say that, and in future assignments, we may also have some repeated calculations. So you only need to show some key steps, or maybe only show detailed calculations for the first one. And you don't have to show every detailed steps. And it is okay if you can write the code, or you can use the Excel to help you to do the calculation. So I think this can save some of the time. And also, Maria will be grading assignments 3 and 4. So if you have any specific questions, you can ask Maria. And so for the next week, everyone is going to watch the 5 videos of the graduated from the, I think 5 graduated students. And so each of you will prepare 2 questions for the video, and you post it on the discussion. So when the guests come, we know what we are going to discuss. And it will count it as the participation. And so do you have?
Yeah, hi guys. I just wanted to stop in and say, please post your data from yesterday's lab in the collaboration doc. Let Laura know if you have any issues accessing it. I did do a